engineer and faculty scholar here at UCSB. This summer I've worked with my mentors Jean Judd and Scott Carmichael and Professors Scott Shell and Patrick Doherty on engineering novel self-assembling peptide materials via simulations of diphenylalanine. So as many of you may know, nanostructured materials are critical to many technologies emerging today. However, if we want to be able to produce these materials on a large scale, we need to have a very reliable, controllable process for their manufacture. Self-assembling peptide materials can form a wide variety of structures, including nanotubes, nanovesicles, etc. They're cheap and easy to make, and are also environmentally friendly. They can also be easily manipulated using known biochemical techniques. Diphenylalanine is one of the first peptides that exhibited this self-assembling behavior, and its molecular structure is shown here on the bottom. The only thing you really need to know about this molecule is it is amphiphilic. That is, it has both polar charged groups here and here, and nonpolar hydrophobic groups here and here. When placed in water, diphenylalanine forms nanotubes, as shown in this SEM image above. These nanotubes are stable, strong, and have many unique properties, including photoluminescence and strong piezoelectric activity. And diphenylalanine nanotubes have many applications outside of the biological field. For example, silver nanowires can be grown on the inside of these nanotubes, and the nanotubes can subsequently be degraded away using enzymes, leaving a discrete silver nanowire. Diphenylalanine nanotube forests can also be grown on a surface using a vapor deposition technique. In this way, we can increase the electrode surface area and thus the performance in supercapacitor devices, which are potentially the future in energy storage. In addition to having many unique applications, diphenylalanine nanotubes also have an extremely unique crystal structure that is shown here on the bottom. So I'll walk you through what this crystal structure is. This yellow tube here is a cross-section of a diphenylalanine nanotube. If we zoom in on these little black dots that are dispersed throughout, we find that they're actually pores, which fill with water when the nanotubes form. If we zoom in on an individual pore, we find that it is made of 6 diphenylalanine peptides, where the peptide is represented here, with their hydrophilic groups facing the inside of the pore where the water is, and their hydrophobic groups facing the outside of the pore, where they stack together. Because this is such an unprecedented structure, especially in biomaterials, there are many questions about how it forms and what makes it so stable. Using atomistic simulations, we hope to discover the driving forces, especially for early self-assembly, the reason for this particular crystal structure's stability, and the overall mechanism and pathway for diphenylalanine nanotube self-assembly. To study these systems, we perform all-atom simulations. An all-atom simulation is simply a simulation in which we explicitly represent every single atom and their physical interaction. This includes not only every atom in the peptide, but also every atom in the surrounding water molecules. For this reason, we are limited to very small-scale systems. To give you an idea of one way we can interpret our results, I show you here a video clip of one of our simulations that we performed, and I just removed the water for clarity purposes. Another way we can look at the results of our simulations is using a contact map. A contact map is made by comparing all possible peptide pairs in the system and counting the number of times non-hydrogen atoms come close to each other in the simulation. In this way, we can get a quantitative metric of the strength of relative interactions between different atoms on interacting peptides. When looking at this contact map, the axes here on the left and bottom are labeled with numbers that correspond to atom numbers on this representation of the diphenylalanine peptide below. Where blue numbers represent nitrogen atoms, black numbers represent carbon atoms, and red numbers represent oxygen atoms. Looking at the contact map, interactions are rated on a scale of red to blue, with red being the highest amount of interaction and blue being the lowest amount of interaction. The first thing we wanted to do was use this technique to look at the crystal structure and evaluate the places in which we saw the most interaction in the crystal structure. The first thing we wanted to look at was this hexagonal pore. It has previously been suggested that this is the first motif seen when diphenylalanine nanotubes form. So we looked for electrostatic interactions between the charged ends, and that corresponds to interactions between the 0 atom here and these atoms 20, 21, and 22, which corresponds to these areas circled in gray on the contact map. What we find is that while there is some degree of interaction between these charged ends, they don't represent the strongest amount of interaction between the peptides. Rather, the strongest degree of interaction is seen here, which corresponds to these so-called second rings on the peptides. Looking at the crystal structure, it's actually not particularly surprising that these are the strongest interactions, as these rings come together in groups of 3, as is circled on this diagram of the crystal structure on the left. We also see a high degree of interaction here on the contact map, and this corresponds to interactions between the first rings on the peptide, which correspond to groups of 2 rings coming together in the crystal structure. For this reason, we conclude that the strongest forces stabilizing the crystal structure are hydrophobic interactions rather than charged interactions. The next thing we wanted to do was allow these peptides to float freely in water and see what happened and see if they formed any aggregates that resembled the crystal structure. We did this by simulating 12 free-floating peptides in a box of water. What we found is that the strongest area of interaction on this contact map is between the charged ends of the peptide, which contrasts from the crystal structure a little bit. When we looked through our videos of our simulation to find snapshots where these structures became apparent, we found that hydrophilic interactions, or charged end interactions, were most important in the formation of small oligomers, or the structures that form when 2 or 3 peptides come together. Looking at this trimer structure here, we see that it forms a ladder-like structure that is connected by these charged interactions, which are represented by the dotted lines, the red dotted lines here. While it's no longer the most prominent interaction, we also see a relatively strong amount of interaction between these second rings, as we saw in the crystal structure. When we looked for snapshots representing this behavior in a video of a simulation, we found that these ring interactions became more prominent as larger aggregates formed, as shown in this cluster of 6 peptides here, where the side chains are these purple groups coming together. The next thing we wanted to evaluate was the effect of local concentration on peptide aggregation. What we find is that as we increase from a low concentration to a high concentration in our simulations, the side chain interactions start to change, from first ring interactions to second ring interactions. This tells us, we conclude from this data that as we increase the local concentration of the peptide, it starts to more strongly resemble the crystal structure. We've learned many valuable things from our simulations so far. Firstly, we have found that the crystal structure shows the most amount of interaction between hydrophobic groups, leading us to believe that it is stabilized principally by aromatic and hydrophobic interactions. From our simulations of free-floating peptides, we have learned that the early self-assembly of diphenylalanine is driven by the amphiphilicity of the peptide. Very importantly, we also learned that this hexagonal pore-like structure seen in the crystal structure is actually not the first structural motif we see when 6 of these peptides come together. Rather, hydrophobic interactions drive the earliest formation of 6-membered oligomers. Additionally, we find that as we increase the local concentration of the peptide, we see similarities with the crystal structure increasing. In future, we'd like to simulate larger systems to observe larger scale self-assembly. I also mentioned that diphenylalanine has been vapor deposited on a surface to form these nanotube forests. So we would like to simulate these conditions as well to determine what the mechanism for this self-assembly is. And even further in the future, we would like to identify novel variants of diphenylalanine that could potentially show increased stability and also form different nanostructures, and then test them experimentally. I would like to thank the Arnold and Mabel Beckman Foundation, and specifically the Beckman Scholars Program, for the funding that made this research possible. Professor Scott Shell and Patrick Doherty, my mentors Jeeheen Jen and Scott Carmichael, and the whole Shell group shown here. And Erica Lubin for all of her support of my research endeavors, both last summer in the SEAM program and this summer in the Beckman program. Thank you.